So, we're diving into a really fascinating outtake on tissue regeneration. It's like um, our skin and other epithelial tissues bouncing back after extreme
Yeah,
damage, right? The study unveils these special cells that sort of defy the usual cell death, almost like a cellular phoenix.
right. You're talking about those DARE cells, correct? They manage to survive even when the death cues are on full blast?
Exactly. Um, so these DARE cells, after irradiation, for instance, in fruit fly larvae, kick in by halting the full-blown action of the caspases.
Mm-hmm.
Their survival lets them multiply and kickstart the tissue renewal process.
And then there are the NARE cells too, right? But they don't activate caspases at all, though they still contribute to repair?
Yes, that's right. It's like, um, imagine you've got a team where DARE cells serve as the initiators. They not only survive the dangerous signals, but also send out growth cues to their neighbors.
Yep.
Meanwhile, the NARE cells, which don't go through the caspase episode, help by balancing the growth so that regeneration doesn't become uncontrolled.
That balancing act is really smart. But don't you think that using the very machinery that usually causes cell death could be a risky proposition? Couldn't this mechanism also be exploited by cancer cells to become more resistant?
Interesting point. And actually, that's one of the study's highlights. The same caspase tethering process that helps tissue bounce back also might set the stage for tumor cells to dodge death, making them like 7 times more resistant
Right.
after subsequent radiation. That duality opens up major opportunities for both wound healing therapies and understanding cancer recurrence.
It's like a double-edged sword. These mechanisms drive healing, but also, unfortunately, could backfire when it comes to tumors.
Exactly. The researchers uncovered that when you remove DARE cells, the whole compensatory proliferation, the tissue's self-repair mechanism, just stops. And they
Mm-hmm.
found that a molecular motor plays a key role by tethering the initiator caspase to the cell membrane, preventing it from pushing the cell all the way into apoptosis.
So, could this knowledge really be translated into human therapies? Improving wound repair while also possibly preventing or at least understanding why recurrent tumors are more aggressive?
That's the exciting part. The feedback loop between DARE and NARE cells ensures that the regeneration remains orderly, avoiding overgrowth.
Yeah,
And this insight, originally spotted in flies, has huge potential for advancing our approach to human tissue repair and possibly fine-tuning cancer treatments that currently rely on triggering cell death.
but isn't it a bit challenging though? I mean, translating findings from fly models to human biology always comes with its set of hurdles.
True, and that's a valid critique. There are complexities when scaling from fruit flies to humans, yet historically, fly models have paved the way for major biomedical breakthroughs. The
Uh-huh.
hope is that with further research, we can harness these regenerative insights without inadvertently fueling cancer resistance.
Really, it's a remarkable balancing act between life and death at the cellular level.
Exactly. And these findings open up avenues for new therapies that, you know, could speed up beneficial regeneration of tissues and maybe even help curb cancer
Yeah,
relapse. It's a magnificent step towards solving a 50-year-old mystery of how tissues regenerate so powerfully after extensive damage.
absolutely. It's not just about mending what's broken, but understanding the underpinning mechanisms that could revolutionize our approach to treatment.
Precisely. And while there's a lot to unpack, it's pretty exciting to think of the clinical potential here.